Hello, welcome back everyone. Magandang umaga sa inyong lahat. So this is the second part of our review. We're going to talk about microbiology and immunology. Hopefully you can see my pointer here. All right, yes. Let's begin. So microbiome, right? So we have in our body some commensal flora or microbiota. It varies anatomically. In your skin, you have Staphylococcus. In your large intestine, you have E. coli. However, blood in CSF, your CSF is sterile, right? So what's the function of your microbiome? So microbiome, it blocks access to others. So your normal flora, it occupies a specific receptor that blocks access to others. Second, your normal flora, they inhibit damaging substances. For example, they lower your local pH, right? Or they are bacteriocins. Another characteristic is the normal flora, they compete more effectively for resources, thereby limiting the availability to your pathogens. Next, characteristics again, protection against disease. However, your microbiome has a possible source of infection. Your endogenous E. coli, if they multiply rapidly, it might cause disease. Another function, barrier, immune modulation. They influence your host metabolism and drug interaction. What if microbiome is gone in your body? What happens? It leads to a loss of diverse beneficial species, which we call dysbiosis. Okay, dysbiosis, loss of diverse beneficial species, as can be seen in someone who has chronic inflammatory disease, your IBDs, inflammatory bowel diseases, your Crohn's disease, and ulcerative colitis. Let's talk about the basic biology of bacteria. Differences between eukaryotes and prokaryotes. Focus on the prokaryotes, high yield. Prokaryotes, cell wall. You have a lipid bilayer membrane. What kind of ribosomes? 70S ribosomes. What kind of genome? Single linear genome. And you have plasmid. What kind of chromosome do prokaryotes have? Either circular or linear. No membrane-bound organelles for your prokaryotes. What's the function of the plasmid? The plasmid, extrachromosomal DNA, it is circular. What's the importance of the plasmid? High yield. It codes for antibiotic resistance, for toxins, and for mobile gene transfer for conjugation, right? The formation of your sex pilus, your mating bridge. Bacteria growth, they can reproduce via what process? High yield binary fission. The growth is exponential. It is measured in your logarithmic of 10. So just if you have one bacterial cell of E. coli, the population doubles. For 20 minutes, it gives rise to 10 million cells, right? In 8 hours. So the bacterial growth via what? Binary fission. What can we expect about the growth? Exponential, meaning it is measured in the logarithmic of 10. High yield bacterial growth curve. So if you put the bacteria on an agar plate, you can have phases, different phases, right? In a closed system, nutrients and space are finite. There's no removal of your waste products. Therefore, the growth rates are variable based on bacteria and conditions. So there are different phases: lag phase, logarithmic phase or exponential phase, stationary phase, and then your death phase. Lag phase. So this is high yield bacterial growth curve. Lag phase, the newly cells, they adjust to your new environment, right? They just adjust there. They are not multiplying at the maximum rate. They're getting acquainted with the agar plate. Next, B, you have the logarithmic phase. The logarithmic phase, from here to here, you can see an increase of growth at an exponential rate. Here, the cells reach maximum rate of your cell division. Is this infinite? the logarithmic phase? No, it is finite. It continues as long as nutrients and environment is favorable. Next, we have the stationary phase. Stationary phase, this is when the population reaches a maximum numbers. Or in other words, the rate of cell inhibition or death equals the rate of multiplication. And the last phase is death phase. We have a decline in growth rate. Why? Depletion of nutrients, of course. The nutrients are finite, right? So, and of course, you increase the metabolic waste products of the bacteria. And you also have limited space there. So, how does the bacteria grow? They need essential nutrients. So you have macronutrients and macro elements. So these are your macronutrients. You can read that one. Macro elements, and you have the trace elements. Bacteria also utilize energy. They need ATP via two process. They can generate ATP via aerobic respiration, or if without oxygen, via fermentation. So aerobic respiration occurs in the presence of oxygen, right? Oxygen is essential to produce ATP. If there's no oxygen, they can do anaerobic respiration. It is less efficient than aerobic. Or they can also do fermentation, right? For example, E. coli, highlight that one. E. coli, it ferments lactose and glucose. So clinical relevance of this. So the microbiota, they are critical for breaking down metabolites for us. And these metabolites can serve as indicators of infection. For example, you can do a nitrate test on the urine, right? If there is E. coli in the urine, you can expect conversion of nitrate to nitrite. So if you check the urine sample, there's a nitrite positive test. It means E. coli. There's E. coli in your urine. Also, bacteria can deplete oxygen in your tissue, causing infection, right? You have anoxic conditions. For example, they can do host tissue damage. They can attack your immune cells. And also, it has antibiotic resistance. So antimicrobials are less effective now. So different growth factors, of course. Physical factors, they affect microbial growth. Temperature, right? Minimal, maximal, optimal temperature. However, this one is a much more high yield slide. Put a star here. So temperature, most are mesophiles. Body temperature, most pathogens are mesophilic. Psychrophiles, they are negative, right? Colder. They grow well in colder temperature. Thermophiles, they grow well in higher temperature. Hyperthermophiles, they grow well in increase in temperature. You can see hyperthermophiles, for example, in hot springs, right? Growth factors, of course, chemical factors. They also affect microbial growth. Oxygen plays an important role. Don't forget, oxygen is a terminal acceptor in your electron transport chain from last term. It serves also as an important regulatory signal for your virulence factors. Of course, you need enzymes required for oxygen survival. Put a star here. High yield. Obligate aerobes, obligate anaerobes, facultative anaerobes. Obligate aerobes, they only grow in the presence of oxygen. Only grow in the presence of oxygen. Obligate anaerobes only grow in the presence of not oxygen. So they don't need oxygen. Obligate anaerobes, they don't need oxygen to survive. It should be no oxygen. Facultative anaerobes, if there is oxygen, it's okay, we can survive. If there is no oxygen, it's still okay, right? So either when there's oxygen or not, it's okay. Obligate aerobes, example of your obligate aerobes, you have your Nocardia, you have your Mycobacterium tuberculosis for your obligate aerobes. For your obligate anaerobes, you have your Clostridia, Clostridium perfringens, Clostridium tetani. For that one, for facultative anaerobes, most of your Staphylococcus and your Streptococcus are facultative anaerobes. So as I've said, obligate aerobes, oxygen is necessary, right? They only use aerobic respiration. If there is no oxygen, they die. Example, Mycobacterium, tuberculosis. Highlight that one. Obligate anaerobes, they cannot use oxygen. Therefore, they die when exposed to oxygen. Okay? Highlight Clostridium difficile and other Clostridiums. Facultative anaerobes, they can use both. They can use both aerobic and anaerobic respiration. Highlight E. coli for that one. Microaerophiles, they're peculiar. Why? They require lesser amounts of oxygen, only 5 to 10% of O2. So they're commonly seen in your stomach, right? Example, highlighted, Helicobacter pylori. So when oxygen is used for generating energy, we have two toxic reactive oxygen species that are generated, right? Let's go back to term one. So you have your superoxide radical and hydrogen peroxide. How are we going to remove the superoxide radical? Via an enzyme, highlighted, high yield superoxide dismutase. By combining it with two hydrogen protons to make H2O2 plus your oxygen. And then this hydrogen peroxide that you make, you need to break it down to form H2O and oxygen. So you need the enzyme catalase, right? So when oxygen is used for generation, you have two reactive oxygen species. One, superoxide radical. Two, hydrogen peroxide. How am I going to remove superoxide radical? Via the enzyme superoxide dismutase to convert it to hydrogen peroxide. How am I going to remove hydrogen peroxide? Via the enzyme catalase to make water and oxygen. Okay, so don't forget, in order to differentiate Staphylococcus from your Streptococcus, you need the catalase test. So catalase positive is your Staphylococcus, catalase negative is your Streptococcus. So in eutrophiles, most human pathogens live between a pH of 6 to 8. Sometimes few microbes in your stomach due to decrease in your pH. For example, you have Lactobacillus H. pylori. Acidophiles, they grow at low pH, or Thiobacillus, they love pH of 2. Sometimes they're obligate acidophile, Euglena mutabilis. I remember during my pre-med, one of my classmates, she's doing a research in Euglena. It's a wonderful species. You have obligate acidophile. Or some are alkalinophiles. You have high levels of minerals or salt, pH 9 to 11. So if we're going to grow a bacteria in the lab, we need to know the media. We need to get a media, right? We need to make sure that they grow well in that medium. So it can either be liquid or broth. You can have solid agar, but best of all, it's agar always. Semi-solid. It's either defined or a rich media. And then when you plate them, here is your agar plate, or you put them here, agar deep tube or a broth or an agar slant. You put them in an incubator, right, for 24 hours. And then in the morning, you can see the growth of the bacteria. High yield. Bacteria that require special media are called fastidious. Fastidious. Example, M. tuberculosis. Highlight that one. They need high oxygen and they need a defined media. And it takes weeks for them to grow in a lab. Another high yield point, the difference between selective media and differential media. So selective media, you limit the growth of the others. That's your selective media. Your differential media, it assists with your classification, okay? So for example, the McConkey agar. The McConkey agar allows for the growth of your E. coli and your Salmonella, restricting growth of your other bacteria. So McConkey agar is an example of a selective media. However, if you put a color indicator there, it is now a differential media. Okay, so differential media, it allows differentiation of growing bacteria based on different characteristics. Specifically, if there's a color indicator, if there's a color change, you're using a differential media, okay? Now, put a star here. This is high yield. Common media for bacterial identification, right? McConkey. What is McConkey agar? Differential, right? It has a color change. For example, if it's a lactose fermenter or lactose non-fermenter, if we are differentiating between E. coli and Salmonella, you put lactose in your McConkey, and then you get the results, right? I get the color change. So another one is blood agar. It is selective, rich media with tryptic soy agar. Chocolate agar, this is a heated blood agar. This is, you use this one for Haemophilus influenzae, right? You need iron and cysteine for this. Next, nutrient agar, rich media for non-fastidious bacteria. So you need to know the different examples for now. Next term, it will be much more specific. Different kinds of hemolysis when you plate them. So you have alpha hemolysis, beta hemolysis, gamma hemolysis. Why do I need to know these? Because in order to differentiate your streptococcus, you need to know the area, you need to know the hemolysis. What kind of hemolysis, right? Does it occur in the agar plate? So alpha hemolysis, lysis of red blood cells with incomplete breakdown. You can see here, there's an incomplete breakdown of their hemoglobin. You can see a greenish, a tan zone here for your alpha hemolysis. Next, beta hemolysis here, complete lysis of your RBCs. So you can see a clear zone around each colony. And then you have the gamma hemolysis. Gamma hemolysis, nothing. There is no lysis at all. Why do I need to know hemolysis? To differentiate Streptococcus species. So put a star here again. Lowenstein-Jensen agar for Mycobacterium tuberculosis. Lowenstein-Jensen, Mycobacterium tuberculosis. Thayer-Martin agar, Neisseria gonorrhea. This is a chocolate agar. Okay? So LJ, Mycobacterium tuberculosis. TM, Neisseria gonorrhea. How are we going to collect specimens? You could also do throat swabs, lots of commensal flora, specifically for Streptococcus infection, for strep throat, or you could do a sputum sample. Sputum sample, right? If for example, for TB is suspected, right? Or you can do bronchial aspiration or transtracheal aspiration. Next, blood. You can also do blood. You can also collect samples via blood, sterile in healthy individuals. Don't forget the presence of bacteria in the blood is known as bacteremia, also called blood culture positive. Septicemia, on the other hand, it is a hypersensitivity or reaction involving different systems in your body. So how are we if we're going to get specimen from the blood, what's the best way to prevent septicemia or bacteremia? Use a sterile technique. Disinfect the skin where the sample is obtained. CSF, it is sterile, normally sterile, right? However, if there's an infection, it can cause meningitis, inflammation of the meninges, encephalitis, inflammation of the brain tissue, or both, meningoencephalitis, inflammation of both the meninges and your brain tissue. High yield, don't forget CSF. It is a stat specimen in the lab if you suspect someone has meningitis or encephalitis or both. Lumbar puncture for your CSF below the level of L4 and L5. Don't forget from term 1, the layers that you need to penetrate to get to the subarachnoid space to get that CSF. Wound specimen. Of course, we can aspirate. In the wound, we collect pus by needle and a syringe rather than a swab, right? This is very important when we suspect what? MRSA, methicillin-resistant Staphylococcus aureus. We're going to learn this one next term. Sometimes we do stool sample, right? So it has lots of commensal flora, so we need to differentiate. We need a medium that is both selective and differential, so we use McConkey agar that selects, that limits the growth of other bacteria and differentiates the classes of the bacteria. Okay, we can also use urine, for example, right? Don't forget, urine is contaminated by commensal microflora. It is collected by what? Clean catch midstream urine, okay? Clean catch midstream urine. Those words that are highlighted on the slides are high yield, okay? So if you have a urine culture, you have three parts. You have a colony count, you can isolate them, and you can have antimicrobial susceptibility testing. How are we going to do this? By getting a calibrated loop, plate them in an agar, and then count the colony forming units. What is a colony forming unit? It is a way to estimate the number of bacteria that are present in your specimen. So you need a calibrated loop to inoculate the entire surface to your agar. So for urine specimen quantification, high yield more than 10 raised to 5 CFU per ml is positive. You have a positive urine culture. Your urine is invaded by a bacteria. In addition to the calibrated loop, you can also do spectrophotometer or live count serial dilutions. What's the benefit of a live count serial dilution? It is more important, of course, for testing growth rates and the curves. So you have a full strength stock here, 1 stock per ml. We actually reduce the concentration. For each reduction, we are reducing the concentration. And then you plate them on an agar plate. You drop a suspension, place onto the central agar plate, and spread over the surface. So you pipette into plate, you can dip the spreader into alcohol, ignite, and then spread the sample across the agar. You can also do sample dilution. So you have this one. You transfer 1 ml to a test tube. And then from that test tube, you transfer 1 ml to another test tube. And then you transfer 1 ml until it is diluted. And then you plate it to the agar. So how are we going to quantify bacteria? Look for visible colonies, right? The assumption is 1 colony arises from 1 bacterial cell. And then you count. When you count the colonies, you call them CFUs, colony forming units. So let's look at this, bacterial quantification, right? Let's look at 10 raised to negative 1, 2 dilutions, third dilution. You can see you have confluent growth. You cannot count. Can you count that? It's too hard to count. It's too much, right? But let's look at the 4th dilution here. The 4th, you can count, yeah? It has 200. So between 30 to 300 colonies for accurate count. For the 5th and the 6th ones, well, it's too few to count. How are we going to calculate? High yield, number of colonies divided by dilution counted times the volume of the drop. Know this formula, please. What's the relevance of counting CFUs? They can quantify bacterial load, meaning how much bacteria is present in a sample, for example, before and after drug treatment. But in order to make sure what kind of species that bacteria is, you need microscopy to identify the actual organism itself. Another way to identify bacteria, right, is using gram stain. So what is this? First, heat fix the bacteria on a glass slide. Then you stain it with a purple, crystal violet. Then you destain with alcohol. And then you have a counter stain with pink saffron. Okay? This is high yield. Gram positive, they keep the purple stain. That's why they look purple. Gram negative, they lose the purple stain and they accept the counter stain. That's why they are pink. And the next slides we're going to talk about why do they keep the primary stain and reject the counter stain. So in other words, This is high yield. Gram-positive, they keep the purple stain. Gram-negative, they absorb or keep the counter stain. This is a beautiful example of a gram-positive organism, your Staphylococcus aureus. This is another example of gram-negative, or your E. coli. You can see gram-positive, purple, gram-negative, pink. What's the stain here? Crystal violet for gram-positive. What's the stain here? Safranin for your gram-negative. You can also classify bacteria based on their shape. Some are shaped like a sphere, pairs and singles, right? Diplococci, we call them. Chains, streptococci. Clusters, staphylococci. Sometimes they look like rods, bacillus, either in pairs or singles, either in chains, sometimes flagellated bacilli. Sometimes they look like a spirochete. Borrelia, Burgdorferi, Lyme disease, Treponema. Look at this one. Treponema pallidum, syphilis. You can also have Spirilla. Now, for the cell structure, right? We talk about cell membrane. What's the function? The cell wall, high yield, it is peptidoglycan layer. Cell wall is peptidoglycan layer. You can also have capsules, appendages, flagella, and your pili. Now, put a star here. This is high yield for the bacterial cell structure. In the gram-positive cell wall, that's why they retain the crystal violet, because they have a thick peptidoglycan layer. They have a thick peptidoglycan layer, and they keep the crystal violet. For the gram-negative cell wall, they have a thin peptidoglycan layer. That's why they lose the crystal violet. Remember, after crystal violet, you put alcohol, right? And then you do the counter stain, the safranin. So here, the gram-negative, they have a thin peptidoglycan layer. That's why they lose the crystal violet and they retain the counter stain. High yield. Gram-positive, more prevalent on the skin and in the environment, and they are less sensitive to desiccation. Another high yield point. What's the difference in structure of your gram-positive and your gram-negative? In the gram-positive organism, you have your LPS. So you have different this one. Sorry about that one. So for gram-negative, organism, you have an outer membrane. You have an outer membrane, and then you have a periplasmic space. For gram-negative, you have an outer membrane and you have a periplasmic space. What is in the periplasmic space? Beta-lactamase. Beta-lactamase is in that space. Why is this important? Next term, you're going to learn about drugs. You're going to learn about pharmacology. We're going to learn about beta-lactamases. So the beta-lactamases, anti-beta-lactamase, they act on the periplasmic space. What is also in the gram-negative? In the outer membrane. In the outer membrane, you have your LPS. You have your endotoxins, right? Your endotoxins, you have your lipid A there, which is antigenic. We're going to talk about this one in the immunology. Next. And then you also have porins there. Porins are just transmembrane proteins that transfer organisms from outside of the outside environment to inside of the bacteria. Okay. For a gram-positive cell wall, think about what's unique in their structure is lipoteichoic acid. They have the lipoteichoic acid, which extends beyond the cell membrane. Okay. So as I've said, the cell wall is peptidoglycan. It is composed of what? NAG and your NAM. What cross links your NAG and your NAM? High yield transpeptidase or penicillin binding proteins. Next term, you're gonna learn about the different drugs the penicillins, the different kinds of penicillins, cephalosporins, monobactams, all of that next term. So this is important. This is a major drug target, your transpeptidase or your PBPs. So peptidoglycan cell wall, it is composed of your NAG and your NAM. What links your NAG and your NAM? Transpeptidase or penicillin binding proteins. What's the significance of your penicillin binding proteins? They are your major drug targets. Here's another example of the NAG and the NAM. Don't forget, the peptidoglycan is the site of action of your beta-lactam drugs, your penicillins and cephalosporins. Also, it is also the site of action of your vancomycin, because the vancomycin attacks D-ALA and D-ALA, the alanine residues. Let's look here. Look at the D-ALA, the yellow one. It links this one, this pentaglycine molecule here. So the D-ALA links this pentaglycine to another pentaglycine, right? And the MAC, the NAG and the MAC, they are on top. You can see here, they are on top, right? And then you have the D-ALA linking the pentaglycine together. So this is the site of your vancomycin. So your NAG and your MAC, in between them, you have your transpeptidases. Transpeptidases here, in between them that links them together, that's your transpeptidases or your penicillin binding proteins, which is the site of action of your beta-lactam drugs, your cephalosporins and your penicillin, which we're going to encounter next term, term 4. So gram-positive cell wall, again, I repeat, thick layer. They retain crystal violet. They have teichoic acid, lipoteichoic acid, okay? What's the function of a lipoteichoic acid? It's a scaffold. You can see here, the lipoteichoic, they extend beyond the layer, right? The peptidoglycan layer. So both molecules, they contribute to the adhesion and recognition by phagocytes. Gram-negative wall, as I've said, so you have a thin peptidoglycan layer. They lose the crystal violet, so they accept the counter stain, the safranin. Characteristics, you have outer membrane. It consists of your lipopolysaccharide. The LPS has, which is an endotoxin. It has a lipid A, which is antigenic, right? And then periplasm, as I've said, this is site of your beta-lactamase enzyme. And then porins, they just transport, cell transport. So LPS is important. Why is it important in gram-negative? Because it has a toxic component of the lipopolysaccharide. So the LPS or lipopolysaccharide, you have 3 components. Uno, lipid A. Dos, core polysaccharide. And tres, you have your O antigen. So lipid A is the toxic effect. This is what the macrophage will gonna identify, the lipid A. Don't forget, lipopolysaccharide functions as endotoxin. Endotoxin, which triggers inflammation, leading to sepsis. So we're gonna learn that endotoxin, it is produced, it is not produced, it is within the cell wall of the bacteria, and this cell wall will be, this component of the cell wall will be recognized by your macrophages and then your neutrophils. It triggers inflammation via IL-1 and TNF alpha. So I'm just integrating immunology to this one. So what's the high yield point here? LPS, 3 components. The most important component, lipid A. That's a toxic component. That's your antigenic component. It functions as what? Endotoxin. What does it do? Triggers inflammation leading to sepsis. So again, put a star on this page. Features, gram-positive and gram-negative, very, very high yield. Next. Another bacterial optional features, you have capsule, you have appendages, flagella, Pili, Fimbriae, type 3 secretion systems, and endospores. Let's go. So what's in the capsule? What is it? What's in the capsule for? What is that? So it's a gelatinous sugar material. What's the function? Resist environmental and immunological insults. For the organism to survive, they need the capsule. So, high yield point. Encapsulated bacteria are not recognized by white blood cells. Thus, the capsule is anti-phagocytic. So, encapsulated bacteria, anti-phagocytic. This is a major virulence factor for your disseminated disease, especially if you don't have a spleen. So, for asplenic patients, you can have increased risk of encapsulated bacteria, your strep pneumo, your Haemophilus influenzae. So they need a capsule to survive. If you don't have a spleen, you are more prone to infections with encapsulated bacteria. Example of encapsulated bacteria, Streptococcus pneumoniae. Another thing that you can see in the bacteria, you have a pili and your fimbriae. What's the function of pili or your fimbriae? For attachment, right? So don't forget the sex pili. They're important for gene transfer, especially in conjugation when you transfer a plasmid from one organism to another organism via a formation of your mating bridge. Flagella. What's the function of a flagella? It's a motor for motility. It's important for classification. Salmonella, for example, is motile. Shigella is not. Salmonella is an H2S non-fermenter. Shigella is fermenter of H2S. Recognized by the innate immune system. So there are different arrangements of a flagella. Monotrichous only on one side. Lopo, one side, but many. Amphitrichous, both sides. Right? And peritrichous throughout the bacteria. So flagella for motility. Salmonella, motile. Shigella is not. Those words in color are high yield. Type 3, secretion system. Only on gram-negative bacteria. So they act as a needle to inject your effector proteins. What is the function of a type 3 secretion system? For virulence factor. For example, the Salmonella, it injects effector proteins. Next, endospores. What's the function of an endospore? High yield again. Highly resistant to desiccation chemicals, hot and cold temperatures. It is found in soil and environment. And you need to know two important genera that form spores: Bacillus and Clostridium. Okay, Bacillus, your Bacillus anthracis, your Bacillus cereus, Clostridium tetani, Clostridium perfringens. Those are important genera. Next, so here we said the gram stain, right? We can identify bacteria via gram staining, via their shape. Look here on the left side. This is gram-positive cocci. This is your Staphylococcus aureus. Here you can see here they look like clusters of grapes, right? Clusters of grapes here, clusters of grapes. So that's your Staphylococcus aureus. On the right side, you can see here gram-negative bacilli. They look pink, right? They look pink like that. This is your E. coli here. Pink, 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 pink. A lot of E. coli. A lot of E. coli. Gram-negative bacilli. Bacilli because they look like rods. Cocci because they look like circle spheres. Aha, this is the dreaded algorithm. Should I memorize this one? Yes. So that next term, you're gonna be all set. So let's approach this one systematically. So gram stain. Is it gram positive or gram negative? So we're looking at gram positive organisms. Gram positive organisms. What kind of peptidoglycan do they have? They have a thick peptidoglycan. What kind of stain they absorb? They absorb crystal violet stain. What's one part of the gram-positive bacteria that cannot be seen in the gram-negative bacteria. Only in gram-positive bacteria you can have lipoteichoic acid. What is the function of the lipoteichoic acid? For stability of your cell membrane, it extends beyond the cell membrane. Good. Next shape. Are they rods, bacilli, or they're spheres, cocci? If they are rods, bacilli, are they aerobic or anaerobic? If they're aerobic, it's bacillus. If they're anaerobic, clostridium. For cocci, are they catalase positive or catalase negative? Do they break down H2O2 to H2O plus O2? If yes, they are Staphylococcus. How are we going to differentiate Staphylococcus? Coagulase. Let's plate them in the blood, like in the sheep's blood, right? If it's coagulase positive, then you have S. aureus. That's your Staphylococcus aureus, or beta hemolysis. They have complete hemolysis of the blood sheep agar. They have beta hemolysis. If they don't, they are coagulase negative, right? I don't know if you've gone into the differentiation of your coagulase negative, right? To your novobiocin sensitive, novobiocin resistant. So novobiocin sensitive is for S. epidermidis. Novobiocin resistant is for your S. saprophyticus. I don't know if you had characteristics of each, but I'm going to give you some tips, okay? S. epidermidis, commonly seen in infections of prosthetic devices. Those who have transplanted knee, those who have prosthetic heart valves, commonly seen, you have S. epidermidis. S. saprophyticus is the most common cause of uncomplicated UTIs in sexually active females. Okay, the most common is E. coli. Right, the second most common uncomplicated UTIs in sexually active females is your S. saprophyticus. For S. aureus, my God, there's a lot to know about S. aureus. They're also oxidase positive, coagulase positive, toxic shock syndrome. It can also cause endocarditis. It can also cause myelitis. Osteomyelitis, I'm sorry. It can also cause skin infection, impetigo, and stuff. So there's a lot. I'm just going to give you an overview, okay? Next, catalase negative. If they're catalase negative, they are strep. And then you divide them via hemolysis. Alpha, beta, gamma. Alpha, strep pneumo. Strep pneumo, optokin sensitive. Optokin resistant, strep viridans. Alpha hemolysis, what do I mean by alpha hemolysis? Partial hemolysis. Next, beta hemolysis, complete hemolysis. Group A strep, strep pyogenes. Group B strep, strep agalactiae. Gamma, no hemolysis. Group D, enterococcus. Let's summarize. Gram stain, gram positive. They can be both cocci and bacilli. For cocci, let's differentiate them via catalase. Are they catalase positive or catalase negative? If they're catalase positive, they are Staphylococcus. Let's differentiate Staphylococcus via coagulase. If they form a zone of hemolysis. If they are coagulase positive, then it is Staph aureus. If not, they are Staph epidermidis or Staph saprophyticus. How are we going to differentiate Staph epidermidis from Staph saprophyticus? Via novobiocin. If they're sensitive to novobiocin, Staph epidermidis. If they're not resistant to novobiocin, Staph saprophyticus. If they don't have a catalase, then they are Streptococcus. Then we need to differentiate them via zones of hemolysis. Either alpha, beta, gamma. Let's go alpha. Alpha, partial hemolysis. Optokin sensitive, strep pneumo. Optokin resistant, strep viridans. For beta, complete hemolysis. Differentiate them via sensitivity to bacitracin. If it's bacitracin sensitive, strep pyogenes. If it's bacitracin resistant, strep agalactiae. Gamma hemolysis, no hemolysis. The only important thing here is group D, your enterococcus, which grows in bile and 6.5 of sodium chloride. Okay, memorize this one for now. It will gonna come handy next term and term 5. Again, gram stain negative. Now they're pink, right? Pink, thin peptidoglycan layer. They have the counter stain. They have the counter stain, the saffron in red. So are the oxidase positive in comma shape? Yes. So you have the differentiation here. It grows in 42 degrees Celsius, Campylobacter jejuni, grows in alkaline media, Vibrio cholera. H. pylori is urease positive. Actually, it's triple positive. Oxidase positive, urease positive, and catalase positive. So for here, you can have diplococci, Neisseria meningitidis, Neisseria gonorrhea. Then you differentiate them via maltose. If they ferment maltose, it's meningitidis. If they don't ferment maltose, it's gonorrhea. If they are rods, the most important thing here is Haemophilus influenza, Bordetella pertussis, Legionella, and Francisella. If they are rods, lactose fermenter and non-fermenter. Lactose fermenter, Klebsiella and E. coli is high yield. Lactose non-fermenter, you divide them into two via oxidase. If they're oxidase positive, it's Pseudomonas. If they're negative, you need another test, urease. If they're negative, check their motility. If it's positive motility, Salmonella, salmon swim. If they're not motile, it's Shigella. If they are oxidase negative, urease positive, then they are Proteus. So I think for now, put a star here. Both of them, learn it now. It will gonna serve you greatly when you're in term 4 and term 5. And for step studying. Okay, but look here. We have atypical bacteria. What do we mean by atypical bacteria? It means they are not related to your gram-positive or gram-negative. They do not respond to first-line antibiotics. They have a unique characteristics. They do not gram stain well. They have a thick lipid wall. They have no cell wall. And they are obligate intracellular. Let's go one by one. Actinomyces. Actinomyces, long branching filaments. Right? Long branching filaments. Two kinds, Actinomyces and Nocardia. Actinomyces israelii. These are your gram-positive branching filamentous. Okay? They are normal flora in your oral and your GI. They form abscess in anoxic tissue. So sometimes you have dental disease, you have a lumpy jaw. They also can cause foul smelling. You can see an abscess here, draining sulfur granules, right? And they can form slow granulomatous abscess. Look at here in this tissue, slow granulomatous abscess. So, Actinomyces israelii, gram-positive branching filaments, commensal, oral and GI flora. They form abscess in anoxic tissue, dental disease, like lumpy jaw, and they form sulfur granules. In terms of Nocardia and your Mycobacterium species, they have this, what we call a mycolic cell wall. They have mycolic acids in their cell wall. They don't have peptidoglycan. They have mycolic acids. The mycolic acids are thick, lipid, waxy layer. So the waxy coat interferes with your normal gram staining. So it doesn't really gram stain well because it's a thick lipid layer. And you also have a cord factor on the surface. Cord factor is important in disseminating your tuberculosis infection. It's very toxic, right? So what's the stain are we going to use? If they can't stain really well in gram stain, what are we going to use? So we're going to use Ziehl-Neelsen stain or acid-fast stain. Okay, acid-fast stain. Why are we using the Ziehl-Neelsen stain or the acid-fast stain instead of the Gram stain? Because they have a thick lipid mycolic acid wall, and when you plate them, when you use the Ziehl-Neelsen stain, they stain red. Okay, high yield. For now, remember, Nocardia is acid-fast, and Mycobacterium is acid-fast. Mycobacterium tuberculosis and Mycobacterium leprae, they are acid-fast bacilli. Next, we'll talk about Nocardia. Nocardia asteroides. These are acid-fast branching filamentous, weakly gram-positive. Well, yes, they are weakly gram-positive, but acid-fast, okay? Don't forget. They are aerobic, and they are transmitted via inhalation of dust particles. They also form abscesses, but where? Pulmonary cavitation is more common. And this is seen in immunocompromised patients. Next, so be sure to summarize the differences between your Nocardia and Actinomyces. Okay, make a table, compare and contrast their characteristics. Next, TB. TB transmission, air droplets. They have a thick cell layer, mycolipid cell wall. They are facultative intracellular, right? They are poorly controlled in individuals with low cell-mediated immunity. If they have low CD4, Th1 cells, they cannot combat M. tuberculosis. They form granulomas, right? Granulomas are formed due to what? Production of cytokines and upregulation of proteins involved in their recruitment. So the macrophages will gonna try to wall off this Mycobacterium tuberculosis in your lungs. That's why they form granulomas. So TB, Mycobacterium tuberculosis, Ziehl-Neelsen stain, acid-fast stain, transmission, air droplets, right? They are facultatively intracellular, more common in individuals with low cell-mediated immunity, low CD4, Th1 cell. They form granulomas. How does it form? Production of cytokines. You have this upregulation of your macrophages. They're trying to wall off this bacteria. However, this can lead to host tissue damage. Mycobacterium leprae, same species as your Mycobacterium tuberculosis. They're obligate intracellular bacteria. They replicate in macrophages and in nerve cells. Transmission is coughing and sneezing. So there are two forms of disease, right? Both causes loss of sensation. So first, tuberculoid leprosy. This is less severe. What kind of T cells is active here? Th1 or Th2? Th1 cell. So CD4 positive Th1 cell, much more active in your tuberculoid leprosy. It's less severe. Your body is trying to fight the infection. The worst one is lepromatous leprosy. Here, you have the CD4 positive Th2 cells are much more active here. So it's now disseminated, right? Lepromatous. Your body is still fighting, but you have Th2 cells. So your body is trying to control, not actively fight infection. This one, it has disseminated already. Lepromatous means it disseminated throughout the body already. So this is deadly, right? Another infection, Mycobacterium marinum. What's the buzzword here? Aquariums or fish. What can we see? Reddish or tan skin with a granuloma. So in the vignette, they're gonna say you have a person who's working on a fish market or aquarium. Then you can see on the skin a reddish tan with a granuloma. Think Mycobacterium marinum. Next, Mycoplasma. What's important about Mycoplasma? They do not have a cell wall. They do not have a cell wall. Mycoplasma. Example, Mycoplasma pneumoniae. This is your atypical pneumonia, your walking pneumonia. They do not have a cell wall. And don't forget, they have a sterols in their membrane. They do not have a cell wall, and in their cell membrane, they have sterols in their cell membrane. Mycoplasma pneumonia, air droplets, as I've said, most common cause of walking pneumonia or atypical pneumonia. Why is it called walking pneumonia? Because when you look at the x-ray, they look worse on the x-ray. Yeah, they have pulmonary infiltrates there, but when you see them clinically, right, they look fine. They look fine, but on the x-ray, they look worse. That's why it's called walking pneumonia or atypical pneumonia. Don't forget, children and teenagers are more prone to get this kind of pneumonia, and it is resistant to first-line antibiotics. Why? Because it doesn't have a cell wall. And remember, the first-line antibiotics, they attack peptidoglycan cell layer, your beta-lactams, right? So since they don't have peptidoglycan layer, they do not have a cell wall, first-line antibiotics doesn't work. Next. So we also have Mycoplasma genitalium and urea plasma. So in women, these are all STIs. So be careful when you go to Grindr or Tinder. So in women, they can cause pelvic inflammatory disease. Or in men, they cause urethritis, right? So urea plasma high yield, it can cause the placenta of pregnant females. It causes spontaneous abortion or premature birth. Mycoplasma genitalium, genital area, right? Genitalium, so STIs, PID, urethritis. Urea plasma, think about spontaneous abortion, premature birth. Oh, we're in the realm of STIs. Right? So chlamydia, obligate intracellular. They are obligate intracellular, meaning they depend on the host cell for energy, for ATP and NAD+. They have a complex life cycle. So you have elementary bodies and reticulate bodies. So elementary body is the infectious form. Elementary body enters the cell via endocytosis. Okay? That's your elementary body. Reticulate body, replicative form. So there are different kinds of species of chlamydia. You have Chlamydia trachomatis for STIs and trachoma, most common cause of blindness. Chlamydia in a newborn. You have Chlamydia pneumoniae by the name itself, pneumonia. Chlamydia psittacosis, most commonly seen in those who work with animals. Chlamydia psittacosis, it's a zoonotic disease commonly spread by birds like parrots, stuff. So, put a star again. Those texts that are in red or different colors are high yield. Chlamydia, obligate intracellular, life cycle, 2. Elementary body and reticulate body. Elementary body is infectious. Reticulate body is replicative. Chlamydia trachomatis for STI and trachoma, the most common cause of blindness in the newborn. Contrast this one with the blindness that you can get in someone who has Neisseria gonorrhea. Here it is again, elementary body and reticulate body, which is infective. Elementary body, which is replicative, reticulate body. So ABC, right? So different kinds of serotypes in your chlamydia. You have ABC, you have trachoma. So you have blinding ocular disease, especially in developing countries. But our focus here is this, D to K. So it can cause this one, cervicitis, STIs, right? Non-gonococcal urethritis, inclusion conjunctivitis. L1, L3 for lymphogranuloma venereum. So what is Chlamydia trachomatis cervicitis? So this is most commonly seen in females, of course. It is asymptomatic. right? Because females only have cervix. So untreated infection can lead to what? Transmission to neonate, pelvic inflammatory disease, ectopic pregnancy, or sterility. As you can see here, this is a healthy cervix. On the right side, you have a cervix infected with chlamydia. How are we going to detect this one? Pap smear. What can we see in a pap smear? Intracytoplasmic vacuoles. If you can see this, intracytoplasmic vacuoles, think about chlamydia. Pneumonia, encephalitis, a typical pneumonia. For chlamydia pneumonia, human to human via aerosols. Chlamydia encephalitis, birds, right? Parrot fever, so zoonotic or occupational hazard. Another obligate intracellular is rickettsia. It is transmitted via arthropod bite, meaning insects. So there are three kinds: rickettsia for Rocky Mountain spotted fever, Prowazeki via a lice, and typhi via a flea. So they penetrate the skin. Once the arthropod bites you, it penetrates the skin or the mucous membrane. It multiplies in the endothelial cells of your small blood vessels. So what if they multiply in the endothelial cells? They can cause vasculitis. You can see here the red structures indicates your histological staining of rickettsia in endothelial cells of a blood vessel. Next, Rocky Mountain spotted fever. After a tick bite, right, you can have a high fever and lymphadenopathy. What can we see? Petechial rash including palms and soles. Petechial rash including palms and soles. You can also have encephalitic sign and gangrene of skin and tissues. All right, I think that's the end of microbiology. Let's begin immunology. So these slides are from my term from last year, so they're kind of old and stale like me. So I don't know if you cover the same materials or not, but no. Please bear with me for a moment. So immunology, right? Immunity, of course, is a function of an individual which recognizes and includes antigenic foreign substances. What's the function? You can read, obviously, defense against infections, tumors, yada, yada, yada. So what's the chain of infection? You have a microbe. The microbe will gonna give you infectious dose. The microbe has virulence factors. It will transmit this to you via what? Via a vector. What kind of vectors? It can either be airborne, it can either be a contact, and then you're the host. You now have the infection. What happens? You have different factors involved: innate immunity and adaptive immunity. So what's the function? They evolve to protect you, right? So you have a pathogen, something that causes a disease, and then an antigen, your lipid A from your LPS. This one is recognized by your antibody or T cell receptor. And then you have an epitope, which is a portion of an antigen that is recognized by an antibody or T cell. So how does the body recognize and respond to an organism, an outside organism? One, there's a pathogen. Two, you have the antibodies or T cell receptors, which recognizes your epitope, which is a portion of the antigen that is specifically recognized by your antibody or T cell receptor. Another high yield point, immunogen versus hapten. So hapten is a small molecule. When combined with a larger molecule, it can elicit the production of antibodies that bind specifically to it. So some hapten molecules on their own, they don't have an immune response. On their own, there is no immune response. However, if you have a carrier molecule and you put hapten molecules on the surface, it now becomes a hapten carrier conjugate. Now you can have an immune response. Immunogen is just a substance capable of eliciting an immune response. Please know the terms here, specifically adjuvant, a substance that mixed with antigen to enhance immunogenicity. Different factors in immunogenicity: molecular size, composition, and adjuvant. You also have some host factors here. You have low doses may induce tolerance or fail to stimulate enough lymphocytes. You can just read that. This is a good slide. Put a star here. Factors that influence the immunogenicity of your proteins. So usually, if the size is large, it increases if the protein is large, it increases immunogenicity. Subcutaneous, much better than intravenous or intragastric, right? So function, defense, homeostasis, and surveillance. You can read that. Okay, 2 arms. Innate arm, non-specific. Different components, phagocytic cells, and antimicrobial peptides. So the first line of defense. What's your first line of defense? The skin, of course. You have the lysozyme there, right? You have a high lactic acid and electrolyte concentration in your sweat. In the stomach, you have HCl. You have digestive juices. The semen has an antimicrobial chemical, and the vagina has an acidic pH. In acquired immunity, it develops in response to infection. It takes several days, right? 2 components of adaptive, cell-mediated, and humoral immunity. What's the attribute of this system? It's specific, right? They can distinguish between 2 proteins. They are diverse. They have memory. And they can recognize self from non-self. High yield, put a star here. This is important, a comparison of innate and adaptive immunity. So we talked about host defenses. Acquired is specific, right? This is your B lymphocytes and T lymphocytes. For innate, first line, you have the physical virus, skin, chemical virus, lysozymes, acidic pH. Then you have the genetic barriers. Second line of defense, inflammatory response, interferon, phagocytosis, and complement. So the acquired is specific, it's responsive, and it is specific. For innate or non-specific, it's pre-existing, present at birth, and non-specific. So cells for each and every one. For adaptive, B cells, T cells, innate, neutrophils, eosinophils, basophils, mast cells, NK cells. So let's look at the cells of the immune system. Polymorphonuclear cells. So PMNs, they have a lobe nucleus. Your white blood cells, the most abundant is neutrophil. There are others, eosinophils, basophils, and mast cells. So let's look at the neutrophils. Neutrophils are multi-lobe. They have 2 to 5 lobes. They're the first to arrive at the site of inflammation. So if you have an increase in neutrophil, it means you have indication of infection. So they destroy the organism via phagocytosis. They are very short-lived, however, only 10 hours in the blood and 1 to 3 days in the tissues. So they come out of the blood vessels to kill. It would cause us too much damage to tissue if they are long-lived. Eosinophils, on the other hand, they are bilobed nucleus located mainly in tissue. This is like a review from term 1, right? Yes. So phagocytize, you have specific granules. Remember, basic proteins, neurotoxin, you have histaminase, peroxidase, cathepsin. What's the function? High yield, parasitic helminths, and allergic reactions. Basophils, you have basophilic granules, heparin, histamine, you have peroxidase. They play a role in what? High-yield allergic reactions. Mast cells, similar to basophils, they play a role in what? Again, if they are like basophils, then they play a role in development of allergies. Leukocytes. Leukocytes are your one-lobe nucleus or your mononuclear leukocytes. They don't have granules compared to your granulocytes, or neutrophils, eosinophils, and basophils. Monocytes are macrophages. They circulate in the blood, and when they migrate, they become macrophages. Different names in different parts of the body. So here it is. You can see here, monocyte, and then they form phagosomes in the tissues as macrophages. They have pseudopodia, and they become macrophages. So what's the function of a macrophage? They survey the tissue, they eliminate, ingest microbes or dead or injured cells, and they extract immunogenic information from those foreign matter. Here you can see a macrophage. Macrophage, they have toll-like receptors there. And then the toll-like receptor will gonna discover a foreign molecule. We're gonna bind to a pathogen-associated molecular patterns, PAMPs, which causes the macrophage to secrete cytokines, interleukins, and inflammatory mediators. Dendritic cells, they express CD14 plus, right? What's the function? Surveillance, inflammatory response, and they are also APCs, don't forget. They are antigen-presenting cells. So here it is. They all come from what? Hematopoietic stem cell. The hematopoietic stem cell divides into two. Lymphoid progenitor, myeloid progenitor. Lymphoid progenitor becomes your natural killer cells, T cells, B cells. Your myeloid progenitor, it divides into two. It can become megakaryocyte, which produces your platelets, and the erythrocyte progenitor. And then the granulocytes, it can become neutrophil, basophil, eosinophil, and monocyte or macrophages. So don't forget dendritic cells, they are antigen presenting cells. So put a star here. This is a good summary slide of the cell type and the principal functions. Lymphocytes, B lymphocytes and T lymphocytes. B lymphocytes, humoral immunity. T lymphocytes, cell-mediated immunity. Antigen presenting cells, they present antigens, of course. Dendritic, macrophages, and follicular dendritic cells. Then you have the effector cells. You have the T lymphocytes, macrophages, and your granulocytes. You can read this one. So put a star here. This is high yield. Another high yield point. Put a star here. Antigen presenting cells, they are ready. What do? They present antigens. They present antigens to your macro, right? So professional APCs are your dendritic cells and macrophages. They present the antigen to your T cells. Now, let's look at MHC class 1 versus MHC class 2. MHC class 1, what kind of cells express, what kind of T cells express MHC class 1? CD8, right? CD8 T cells express MHC class 1. What is unique in MHC class 1? Beta-2 microglobulin. Beta-2 microglobulin. For MHC class 2, CD4 cells. T cells express MHC class 2. What's unique in MHC class 2? You have 2 alpha and beta. See? 2 alpha, alpha 1, alpha 2, and beta 1 and beta 2. Lymphocytes, different kinds of lymphocytes. Natural killer cells, B cells, and T cells. Look, they are agranulocytes. They don't have granules compared to your neutrophils, eosinophils, and your basophils. Put a star here. This is high yield. So T cell, B cell, natural killer cells. Natural killer cells, they perform immune surveillance, right? B cell, this is the one that produces your humoral immunity, right? They produce your immunoglobulins, your antibodies. T cell divides into 3. You have cytotoxic T cells, CD8 T cells. They become helper T cells, your CD4 T cells, or they become suppressor T cells. These are your regulatory T cells. So lymphocytes again, surface markers CD16 and CD56. Now we go to the meat of the matter, the B and T cells. So the B lymphocytes, the site of maturation is in the bone marrow. So in the bone marrow, once antigen is encountered, right? So they're circulating in the blood and they encounter an antigen. So what happens? They differentiate. They become plasma cells, right? And then they become memory B cells. For the T lymphocytes, the site of maturation is in the thymus. So the only recognized antigen that is bound to your cell membrane proteins, or MHC, okay? The only recognized peptides bound to MHC. Different kinds: effector T cells, memory T cells. You have different effector T cells: T helper, cytotoxic, and your T regulatory cell. This is a good picture, but let's check here. T helper cells. So T helper cells, CD4, right? Your CD4 T cells, they recognize antigen presented by what? MHC class 2. MHC2, CD4. Cytotoxic T cells, CD8 cells. What's the function? Recognize antigen in MHC class 1. Class 1. Remember the rule of 8 from first aid? 1 times 8, 8. Here, 2 times 4 is 8. Right? The rule of 8. So for cytotoxic, they are good in eliminating viral or intracellular microbes. So here's just a picture. You have the antigen here, MHC class 2. It recognizes the T cell receptor. This is the CD4. When that happens, you have a release of cytokines from the T cell receptor. This is a cytotoxic T cell. T regulatory cells. What are the CD markers? CD4, 25, and FOXP3. So Tregs, they help suppress the immune system. They maintain tolerance and prevent autoimmune diseases. Memory T cells. So memory T cells are there after they have previously encountered and responded to an antigen. They can reproduce to mount a faster response. They can either be CD4 or CD8 positive. Put a star here. This is high yield T cell types. Look at the T helper cells, the CD4. 2 kinds, TH1 and you have a TH2. T regulatory cells, all right, they are suppressor. They suppress the immune system. CD4, CD25 on their surface. What cytokines do they produce? TGF beta and IL-10. Beta, IL-10, reduce immune system. Cytotoxic T cell, right? CD8 on the surface. They release what? Mediators of inflammation, your perforins, granzymes, and your fast ligand. T helper cells, 2 kinds, TH1, TH2. TH1, they produce what? Interferon gamma and TGF beta. TH2, they produce what? IL-4, IL-6, IL-10, and others. Please know the kind of T cell, the function, the surface markers, what MHC is expressed, and the cytokines or mediators that are expressed by each cell. Those are high yield. Put a star here again. This is a good table that summarizes the cell type with their appearance, the function, and some high yield comments there. Okay, we're talking about CD. What is that? That's old, right? We now have Blu-ray, right? We no longer have CDs. So cluster of differentiation is, you need to investigate. They are expressed in the cell surface, right? So they are established in actually 1982. They are assigned based on a one cell surface molecule recognized by 2 monoclonal antibodies. So let's look at the table. Stem cell has what? CD 34, 34. Stem cell. It produces 1, 2, 3, 4, 5. A granulocyte, monocyte, T lymphocyte, B lymphocyte, and a thrombocyte. They all have what? CD45, CD45. They all have CD45. Now, we need to differentiate between each of them. What do they have differently? They all have CD45, but in granulocytes you have CD 15, right? 15. Monocyte has CD14, 14. T lymphocyte, you have 3, CD3. B lymphocyte, you have 19, CD19. And thrombocyte, you have CD61, 61. Again, stem cell has CD34. The stem cell will gonna produce 5 lineages. 1, 2, 3, 4, 5. We need to differentiate them. All of them has CD45. Granulocyte has CD15. Monocyte, CD14. T lymphocyte, CD3. B lymphocyte, CD19. Thrombocyte, CD61. How do I remember T lymphocyte from B lymphocyte? T is for toddler. So CD3, like CD3, 4, 5, 6, 7, 8, 9. So those are T lymphocytes. If you have CD 11, 12, 13, 14, those are B lymphocytes. Now for T lymphocytes, we need to differentiate them between cytotoxic and T helper. So for cytotoxic, they have CD8, T helper, CD4. And then for this one, you have CD25 for your activated T lymphocytes. Do I need to know the CD number? Absolutely. You need to know the clusters of differentiation in each of them, obviously. Again, put a star here. You have the common CD markers, and where can you find them? So the questions will gonna be what differentiate each, right? So each lineage has a different clusters of differentiation from the other. So memorize the differences. Memorize the differences. CD8, CD4, CD25, 14, 3, 19, 61, 15. Put a star here. This is high yield CD markers on your leukocytes, the differentiation. So hematopoiesis is your production of blood cells. So we're talking about the progenitor cells. We can have the stem cells, and then you can have the myeloid and the lymphoid progenitors. You can read that one. We talk about that. We talk about this one, the red blood cells and white blood cells. And the different leukocytes. There you go. The different image. I think it's good to know the different image. They're gonna show you images. I've seen UWorld questions with images. It's just good to check that. Is this important? I don't think so. Now, don't forget that you have the reticuloendothelial system. These are a network of connective tissue fibers and cells of surrounding organs that are inhabited by your phagocytic cells. They are ready to attack, and they ingest microbes that pass the first line of defense. So we just talked about the different body compartments that participate in the immune system in the blood. You have the mononuclear phagocyte or reticuloendothelial system, the spleen, for example, and then you have the lymphatic system. You can read that one. Let's emphasize important things. So lymphoid tissues. What's the difference between primary and secondary lymphoid tissues? Primary is bone marrow, so B cells, thymus, T cells, right? These are the site of maturation, correct? Development and maturation of the lymphocyte. How about the secondary ones? Your spleen, your adenoids, tonsils. What are those? Those are the places where the mature lymphocytes meet the pathogens, okay? So primary, development and maturation. Secondary, where the mature lymphocytes meet the pathogens. Thymus, T cell development and maturation, right? And it functions decrease with age. Don't forget your favorite disease of thymus, the George syndrome. This is the thymus. You can have immature thymocytes in the cortex and immunocompetent T cell in the medulla. In the cortex, you can have positive selection, and the medulla, you can have a negative selection of your T cells. Lymph nodes, don't forget in the cortex you have B cells. In the cortex of the lymph node, B cells only. Paracortex, T cells. How do I know T cells, paracortex? T cells, paracortex, and dendritic cells. Cortex, B cells, paracortex, T cells, and dendritic cells. In the medulla, you have plasma cells. What is a germinal center? Germinal center is an area of your activated B cells that will proliferate, that will go through somatic hypermutation and class switching. Is this high yield? This is term 1 stuff, so you should know this, the lymphatic flow. So the spleen is a part of the reticuloendothelial system. It filters blood. It is important in your systemic infection. So in the pulse of the spleen, you have T cells. In the follicles, you have B cells, right? In the marginal zone where the antigens meet your APCs, you have phagocytosis of antigen by your innate immune system, and the APCs meet lymphocytes, which is the initiation of your immune response. Spleen, filter blood. Why is this important? It is important in systemic infections. If you don't have a spleen, what happens? Increased risk of encapsulated bacteria. Strep pneumo, for example, Haemophilus influenza. Pulse of the spleen, you can see T cells, follicles, B cells. What's in the marginal zone of the spleen? Initiation of immune response. The antigens meet your antigen-presenting cells. Malt is a secondary lymphoid organ found in your digestive, respiratory, and urogenital tracts. So pathogens are transferred across mucosa by your M cells, the M cells of your Peyer's patches in your large intestine. So malt is just there, right? It lines your digestive, respiratory, and urogenital and tract. What is that organism that invades the M cells? Shigella. So Shigella is an important organism that invades the M cells of your Peyer's patches. Here it is. Now let's look at the second line of defense. We talk about PAMPs and DAMPs. PAMPs, pathogen-associated molecular patterns, and DAMPs, damp-associated molecular patterns. So what happens? So you have now your cell, right? It has a toll-like receptor. The toll-like receptor, let's say this is a macrophage. The toll-like receptor encounters DAMPs and PAMPs. What happens? We have an increase in NFK beta, increased production of NFK beta. We're going to induce now your DNA to produce what? To upregulate cytokines and MHC. MHC, which leads to inflammation, tissue repair, and adaptive immunity. So PAMPs and DAMPs. So toll-like receptors, right? They're the ones that are going to have the phagocytes, okay? So let's look at phagocytosis. What's the purpose of phagocytosis? Ingest and eliminate materials, foreign materials. So you have neutrophils, macrophages, and others. So first, what happens? So you have opsonization, opsonin, C3b, right? So why is there opsonization? Why does it matter? It matters because to ensure that the cell is readily identified. So it enhances phagocytosis. So you need receptors. You need CR1 and then the FC region of your immunoglobulin. So the antibody is still going to opsonize the antigen. What's the main opsonin? IgG. The complement molecules also opsonize the antigen. The most important thing here is C3b. C3b is an opsonin. Okay? So once they encounter this one, once they encounter the foreign organism, what happens with your phagocytes? They ingest it, they form a phagolysosome, and they destroy it via oxygen-dependent system or liberation of lactic acid, lysozyme, and nitric oxide. So what happens? First, you have chemotaxis by phagocyte here. Second, they encounter PAMPs and DAMPs. And then they engulf the organism into phagocytic vacuole. They form a phagosome. And then they combine with a lysosome. We now have a phagolysosome formation. Then they kill and destroy the bacterial cells. And they release via exocytosis your residual debris. So mechanisms of killing: oxygen independent. You don't use oxygen. So example, lysozyme and lactoferrin. If you use oxygen, it is much better, right? So you use oxidative burst. So this is like a review from term 1. Know this, you should know this already. So we're going to kill the bacteria using oxygen. NADPH oxidase, right? NADPH oxidase, it produces superoxide, right? Which recombines with other molecules to produce reactive oxygen species. If I don't have NADPH oxidase, I will have chronic granulomatous disease. So CGD, deficiency of NADPH oxidase, you are susceptible to pneumonia, abscess, arthritis, osteomyelitis, bacteremia, and superficial skin infections. How am I going to test CGD? Nitro-blue tetrazolium test. So in CGD, the test will be negative, yellow. There's no change in color from yellow to blue. It stays yellow. Another disease that's important is Shediac-Higashi. Shediac-Higashi, autosomal recessive. You have a mutation in your LIST gene, a lysosomal trafficking gene. So you're susceptible to infections, anemia, hepatomegaly. Don't forget albinism. Clinical presentation is albinism, neutropenia, periodontal disease, and recurrent infections. So in the vignette, they're gonna put surely albinism there so that you can pick its Shediac-Higashi syndrome. So another question for CGD, right? What kind of organisms you're susceptible to for CGD? Catalase positive organisms. You're much more susceptible to catalase positive organisms. Now next, inflammatory process. Signs and symptoms. We talk about this one in term 1. You should know this already. Rubor, redness, due to what? Histamine release. Tumor, edema, due to what? Histamine release. Where? Post-capillary venules. Calor, heat. Why? Remember, for calor, you have dysregulation of your anterior hypothalamus for term 2, right? Dolor, pain. What causes pain? Bradykinin, PGE2. And then you have the loss of function or functiolyasa. Voila, put a star here. This is high yield. Talked about histamine, talk about bradykinin, BGE2, we talk about macrophages. So what happens? 1, you have a tissue damage. Tissue damage, it causes what? Transient vasoconstriction. And then it causes release of what? Chemotactic factors. The triggers local increase in blood flow and capillary permeability. And then the permeable capillaries, they allow an influx of your exudate and the cells. And then you have the phagocytes. Now they migrate. So from here you have extravasation, your neutrophils and other phagocytes, they migrate to the site of inflammation. We call this one chemotaxis. And then they destroy the bacteria. So unique characteristic of leukocytes, diapedesis and chemotaxis. They migrate out of the blood vessels into the tissues and also chemotaxis. They migrate in response to specific chemicals. Don't forget your cell adhesion molecules, or your CAMs. You have the selectins, mucins, integrins, and ICAMs. Okay, now we go to the nitty-gritty. This is high yield. Put a start here. Inflammation, 4 steps. Rolling, right? Rolling. Well, actually, I'm gonna add another step here. What is that? Margination. Margination. So normally you have a laminar blood flow, right? In your blood vessel. Since you have inflammation, you don't have a laminar blood flow. So the first step should be margination. The neutrophil will gonna go to the margins of your endothelial cell. Second, rolling. Rolling, what is there? So you have your P-selectin and L-selectin. They're like your speed bumps, right, on your endothelial cell. They react with the sialyl Lewis on your macrophages or your neutrophils, okay? So for rolling, you have P-selectin, L-selectin expressed on your endothelial cell. They act like speed bumps. On your neutrophils and monocytes, they have your sialyl Lewis. And then after they roll, they interact together, and then they stop. When they stop, what happens? So your ICAM and the endothelium will gonna react with integrins in your neutrophils and monocytes. Why? So that they can do diapedesis. They can go out of the blood and go into the cell, to the site of the inflammation. And then you have the migration. High yield. Where is the ICAMs expressed? Right? ICAMs expressed in the endothelial cell. Where is the integrin? It is expressed in your neutrophils and monocytes. Where is the P-selectin and L-selectin? It is expressed in your endothelial cell. Where is the sialyl Lewis? It is expressed in your neutrophil and monocytes. So here's just a picture, what we talked about. Another example, high yield, is LAD, deficiency of beta-2 integrin, your CD18. What MHC class molecule did we see beta-2 integrin? We see this one in MHC class 1 for your beta-2 integrin. So this is a deficiency of CD18, right? So what happens? Neutrophils cannot extravasate. They cannot undergo diapedesis and fight bacteria in the tissues. So what can you see in the blood test? You can see neutrophilia in the blood, increase in neutrophils in the blood. Why? They can't go into they cannot extravasate out of the blood vessels. So mediators of inflammation, no bradykinin, right, for pain. Of course, you have the clotting systems here, fibrinolytics, nephrotoxins. Okay, let's look at this one. Put a star here. So you have endothelial damage. What happens? Activation of Hageman factor or Hageman factor. What is this? Factor 12. Where can we see this one? Extrinsic or intrinsic pathway? Intrinsic pathway, right? We're going back to term 1, ladies and gentlemen. So this is Hageman factor. What happens now? You activate the clotting cascade, right? Thrombin from fibrinopeptides in fibrin, and then you have fibrin degradation. When fibrin is degraded, what happens? You increase vascular permeability. You also attract neutrophils. Factor 12 also induces production of prekallikrein. And kallikrein is important. Why? Because it induces formation of bradykinin, which is a peptide. And bradykinin is important. Why? It increases vascular permeability, vasodilation, and pain. And it also induces smooth muscle contraction. Another one, plasmin complement activation. Here's a summary again of what's happening. Don't forget, IL-8, C5a, LTB4 is chemotactic for your neutrophils, right? IL-8, interleukin 8, C5a, this is from your complement pathway. LTB4, where does this come from? Term 1, this is from your arachidonic acid pathway. So acute inflammation, you have increase in IL-1 and TNF-alpha. Inflammation, IL-1, TNF-alpha. Inflammation of neutrophils, IL-8, leukotriene B4, and C5a. Those are high yield. Please know them. Vasodilation, bradykinin, and then macrophages secrete IL-1, IL-6, TNF-alpha. Macrophages always secrete pro-inflammatory interleukins and chemokines. Systemic acute phase reactants, these are your IL-6, right? So, for example, IL-1, it induces fever in your hypothalamus. And then IL-6, it increases production of acute phase proteins on your liver. TNF-alpha also is for pro-inflammatory, okay? So for chronic inflammatory response, it lasts longer than 2 weeks for that. So you have interferon gamma and TNF-alpha, they play a major role. Always associate interferon gamma and TNF-alpha with chronic inflammation. Now, interferons. What's the gist about interferons? So interferons are, for example, I am a cell. I'm infected by a virus, right? I am going to produce interferon to go to the other cell and tell them, hey, there is a virus. There is a virus that's going around. I want you to be immune to that virus. That's the main function of your interferon. They act on neighboring uninfected cells. You're going to learn next term the different kinds of interferon with regards to treatment to different kinds of diseases. So this is viral only, viral, okay? Interferon is for viral infections. You have type 2 there, interferon gamma. Everything that's different in color, that's high yield. Please memorize that. Okay, as I've said, interferon gamma, they form granulomas, right? It's a way that the body, if it cannot remove, for example, in Mycobacterium tuberculosis, you need to form granuloma. So it is activated by Th1 helper cells. So the Th1 helper cells, they aggregate around the macrophages. Your Th1, they secrete IL-1 and IL-12 to induce your macrophages to increase their number, and the macrophages in turn, they secrete interferon gamma. What's the purpose of this? To wall off the infection. Here it is, a good granuloma. Now let's talk about complement activation. So the main thing here is this. So there are 3 pathways, right? So you have the classical pathway, think about C1. You have the alternative pathway, which is automatic conversion. And then you have the mannose binding lectin pathway for bacteria, like mannose binding lectin proteins and stuff. So these 3 pathways, they produce C3. C3 produces C5. C5 produces MAC, membrane attack complex. So that's the gist of it. Now, let's look at this one. As I've said before, 3 pathways, alternative, classical, lectin. You all produce C3, then C5, and then you have membrane attack complex. Classical pathway, you need C1 for your adaptive immunity. Alternative is spontaneous cleavage of C3. Is memorizing the pathway like, okay, this one we're gonna go C4, C2, C4B, C2A? No, it's not. Just get a big picture. 3 pathways produce C3, then C5, then MAC. Okay? No need to be nitty-gritty about the specifics of the pathway. Put a star here. Boop, boop, boop. Classic, alternative, and lectin. Know this table, right? So membrane attack complex. So what's the function of a complement? You can have cell lysis, C5b to C9. This is your membrane attack complex. They're antibody dependent and antibody independent. Opsonization, C3b. B, opsonization. C3b for opsonization. Inflammation, C3a, C5a. A is for anaphylactic, right? Anaphylaxis. So think about inflammation. So C3a, C5a, there's A there. It's anaphylaxis, it's inflammation. Clearance of immune complex, C3b. But the main thing for C3b is opsonization. So what's the importance of this one? Of your pathology, right? So C1, if you have C1 deficiency, if you have deficiency in your C1, C4, and C2, you are more prone to immune complex disease. Example, SLE, you know this one from term 1, glomerulonephritis and vasculitis. If I have a mannose binding lectin deficiency here, you have recurrent pyogenic infection in childhood. Pyogenic means bacterial. Think about bacterial infections. So you have pneumococcal, Neisseria infections. If I have deficiency in the MAC pathway, I cannot produce MAC, then I am prone to Neisseria infection. Neisseria infection for MAC deficiency. If I have a deficiency in C3, I have increased risk of infection with encapsulated organisms. So C1 inhibitor, it can cause deficiency in C1 inhibitor can cause angioedema. That's why you don't give ACE inhibitors to those who have C1 inhibitor deficiency. And for DAF, high yield paroxysmal nocturnal hemoglobinuria, lysis of red blood cells. Put a star here. No hereditary angioedema. You can have subcutaneous and submucosal swelling. How are we going to test? We're going to test via CH50, right? The amount of serum required to lyse 50% of your antibody sensitized erythrocytes. Okay, C1 inhibitor defect, reduce C4, hereditary angioedema type 1. Do not give ACE inhibitors. Okay, now we're talking about antigen receptors of your lymphocyte. So they have variable. So variability, it is concentrated on your complementary determining regions. Now for your B cell receptors, they are your antibodies, right? They're recognized a lot actually. Proteins, lipid, carbohydrates, and nucleic acid. They exist in two forms, membrane-bound and secreted proteins. Now, two regions here is important, your FB and your FC region. For FB, you have antigen binding sites. For FC, you have your complement binding site. Do I need to know the light chain and the heavy chain? Yeah, so light chain, it can be kappa or lambda. Heavy chain, it determines the 5 classes of immunoglobulins. Put a star here. The difference between isotype. When we say isotypes, the classes of immunoglobulins that you can have. Is it IgG, A, M, E, or D, right? Allotype, these are allelic variants. It affects only the constant region. Idiotype, it affects the sequence variability in your antigen binding region. Need to know the difference of the 3. So, put a star here. Classes of immunoglobulins, IgG, A, M, D, E. IgG, most prevalent, crosses the placenta. IgA, dimer, seen in mucus secretions. IgM, commonly seen in acute infections. IgD, doesn't really matter, but this is antigen on B cells. IgE, allergic response, and parasitic worm infections. Put a star here again. This is a great summary. Which one is a monomer? Which one is a dimer? Which one is a pentamer? Right? How many antigens do they bind? So this IgM is a pentamer, so it can bind 10 antigens. Avidity means for the B cell receptors, right? The strength of interaction is affinity. You need to know the difference between affinity and avidity. Affinity is the strength of the interaction between the binding of the antigen to the antibody, while avidity refers to the strength of multiple affinities of individual binding interactions. For example, in 10 for IgM compared only for 2 in your IgG. So affinity refers to the strength of a single antibody-antigen interaction. Avidity refers to the strength of all interactions combined. Okay, and cross-reactivity is an antibody may react with 2 different epitopes. B cell receptor. So how are we going to activate our B cell? So it can either be T cell dependent or T cell independent. So for T cell dependent B cell activation, you have an antigen presentation here by the T cell, then the migration of activated T cells to the edge of the follicle, and then you have B cell activation, the B cell antigen presentation bidirectional, and then antigen uptake and processing. You have the B cell activation. In terms of the T cell receptor, we talked about the alpha chain and beta chain of MHC1 and MHC2. They do not undergo class switching for your T cell receptors. What's high yield? High yield B cell development. Let me see. Where is that BTK gene? So here, for the star, it talks about the progression of your B cell from stem cell to pro B cell, pre-B cell, immature B cell, and mature B cell. So stem cell, you have a germline DNA, right? DNA, then recombination to become a pre-B cell. And then another form of recombination, become immature B cell, and then mature B cell. Look at this one. You have a pre-B cell receptor only on pre-B. Immature B cell, you only have IgM. Mature B cell, you have both IgM and IgD. That's high yield. Put a star on this table. Check the Ig expression. Nothing here, no immunoglobulin, only pre-BCR here and pre-B. Only IgM in immature B, and IgM plus IgD in mature B cell. Another important thing, where is that? Remember your T cell, right? Your T cell positive selection. Where does it occur? Positive selection, it's in the cortex. Negative selection in the medulla. For positive selection, you are testing if the T cell will gonna bind to its own MHC, to its own major histopathocompatibility complex. For the negative selection, if the T cell will bind to self-antigen or not. If they bind to self-antigen, then they're gonna destroy. They're gonna be destroyed via what process? Apoptosis. All right, that's it. Positive selection and negative selection. So another important, put a star here, is AIR, the transcription factor. in the medulla. It drives negative selection of self-recognizing T cells and know the autoimmune diseases involved in your air genes. Positive selection and negative selection in the thymus of your T cell. Positive selection, MHC. Negative selection, self-antigen. If you didn't pass positive and negative selection, you're going to die via apoptosis. Now, for diversity of your lymphocytes, right? It can either be via somatic recombination or junctional diversity. So you have the VDJ somatic recombination, right? They recombine during antigen-independent phase. This is high yield. Put a star here, please. We're almost there. What time is it? Okay, so let's look at the B cell maturation, right? So high yield here. So for somatic recombination, you need RAG1 and RAG2. So RAG1 and RAG2 needs to be expressed in order for your B cell to become an immature B cell. All right? So from lymphoid stem cell to become immature B cell, you need RAG expression. All right? You also need TDT. TDT from pro-B cell to pre-B cell. From a progenitor B cell to a pre-B cell. MHC2 is expressed from pro-B cell to mature. And CD19-2140 is expressed throughout. So high yield somatic recombination RAG1, RAG2. Somatic hypermutation, you need AID. Nucleotide addition, you need TDT. Class switching, you need AID. Do I need to know the exact process of somatic recombination? No, that's low yield. This one, yeah. Just remember, somatic recombination, there's a rearrangement from your heavy chain and your light chain. Now, this is the importance of the TDT that I'm talking about. It adds P and N nucleotides. So TDT, it adds N nucleotides to your so it occurs in heavy chain. So heavy chain is added by your TDT, right? This is very important where in your VJ, VDJ, or VDJ, any combinations, right? For your junctional diversity. So TDT, it adds N nucleotides or repair enzymes and complementary nucleotides. So TDT is there again, as I've said. That's not high yield. So another one is somatic hypermutation. What's the function of somatic hypermutation? This is only for B cells, right? When they migrate to your secondary lymphoid organs. As they proliferate following antigen, point mutation can occur. Why do we have somatic hypermutation? In order to have affinity, to improve affinity. What's the enzyme that is needed for somatic hypermutation? AID, activation induced deaminase. What's the function of AID? It produces diversity in your P cells. What's the exact biological mechanism? Via converting cytosine to uracil. So basically, it's like a mutagen. How do you do class switch? Which goes through class switching. So T cell activation through CD40 and CD40L interactions. Regulated by what cytokines? IL-4, IL-5, interferon gamma also. What does it do, class switching? It allows your VDJ units to be associated with different constant regions. So it only occurs after antigenic stimulation. After they encounter an antigen, you can have class switching. So you don't need to be to know the details, I think. All right, so sources of possible diversity in mouse immunoglobulins and TCR. So you can have here, look at this one, somatic mutation in your alpha chain. beta chain, gamma chain, and your delta chain can induce somatic diversity. Don't forget also the interleukins that are involved, right? The different kinds of interleukins that are involved: IL-1, IL-2, IL-3, IL-4, IL-5, and IL-6, right? IL-1 is for fever, IL-2 is for T cell activation, IL-3 is for B cell activation, IL-4 is IgE, class switching to IgE, IL-5 is IgA activation, and IL-6 is for your acute phase reactants. Now, let me just see. I think that's it. Yes. So if you have any questions, you can just DM me or post on the Facebook page. Any clarifications? If you have first aid, read the biostatistics epidemiology section on your first aid. The immunology section for first aid, I think it's not that in-depth as you have on your slides. If you have Dr. Ramos for immunology, use her slides. She actually highlights all the important things there. All right. Good luck on Monday. You're gonna be great. And stay safe and healthy, and see you all in the future, in the near future. All right. Good luck.